we've known for over a century that there are specific molecules inside our cells that carry information in a 4-part code. And then our cells use groups of that code as a blueprint for how to make the proteins that govern so much about us, from how we look to how we think to how we get or fight disease. But for most of that century, we could only read this genetic code. We couldn't change it.
How what you pioneered with Emmanuel Charpentier was different from the gene therapies and other gene editing systems that came before. What was the big insight, and why is it so important?
CRISPR is a technology that came out of fundamental science. The very first tool that was used for gene manipulation really was coming from the natural world, and that was viruses. Some viruses naturally insert into the human DNA in our cells. HIV is one example of a virus that does that. By studying how viruses manipulate DNA and how they get into DNA, there were a lot of insights about how we could potentially, as scientists, change DNA sequences, but we didn't have the tools to do it. So what happened next was that there were a series of tools developed to find a particular set of letters in the DNA of, say, a human cell and alter them using tools that were generated for that specific purpose. For the wonkier folks in the audience, these include zinc-finger nucleases and TALEN proteins. These showed that DNA manipulation was possible and also could be very powerful. But what was challenging with those tools is that they were bespoke, meaning you had to make one of them for every single change that you might want to produce in a cell, for example. And that meant that they were expensive, they took a long time to produce and then to deploy. And this is where CRISPR comes in, because what viruses and bacteria over eons interacting in nature had figured out is that they could use proteins that were directed by molecules of RNA, which are chemical cousins of DNA. They interact with DNA, so they look at the DNA sequence on a letter-by-letter basis, but they allow easy recoding. And so scientists, once we understood how that recoding works with RNA, we could easily manipulate and change the sequences in DNA that CRISPR can recognize. And that's really the crux of the technology. It becomes a programmable tool that can be directed to essentially any DNA sequence where it can make a change in a targeted way and in a way that is easily controlled in the lab.